Hello, everyone, and welcome to this year's Save Our Seas Foundation Distinguished Speaker Series 2024. We're so happy to have you guys here. This is the first of a whole year of distinguished speakers coming in and sharing their research, sharing their amazing brains with us, and telling us all about what they're doing to help conserve sharks and rays all around the world. And this year, this series, we'll have guests from here locally in South Florida, but as far away as Africa and Indonesia and other places around the world. So we're so happy to have you guys here and to be part of the series. Follow along. Go ahead and register on the MODS website for the whole series so you don't miss any of them. My name is Lance. I'm the Director of Environmental Sustainability at the Museum of Discovery and Science. And our lovely museum is located in very sunny South Florida. If you're new to the area or tuning in from somewhere else, come on by, come and check us out. This is our lovely museum. Our mission there at the museum is to connect people to inspiring science. Everything that we do is kind of driven by four major content pillars: physical science education, early childhood education, health and wellness education. And then tonight, and what I specialize in, is our environmental sustainability. So we have a wonderful speaker for you guys tonight to talk all about their research, specifically white-spotted eagle rays, some of the most beautiful things we have here in South Florida. But before that, I just want to give a shout out to the Save Our Seas Foundation. None of this is possible without them, so we really do appreciate them supporting the speaker series, and also all the researchers that are going to be coming to you guys, including the one that's tonight. So thanks, Save Our Seas Foundation. Congratulations on 20 years of helping the ocean. So that's awesome. Tonight's speaker is Ariadna Rojas Corzo. She's a PhD candidate pursuing a degree in integrative biology, marine science and oceanography at Florida Atlantic University, FAU, here locally in South Florida, in the Harbor Branch Oceanographic Institute. Adi completed her undergraduate studies at the University of Havana with a bachelor's degree in science and joined FAU in fall of 2020 to pursue a doctorate degree, completing her master's degree along the way. She works in the Fisheries, Ecology, and Conservation Lab under the guidance of Dr. Matthew Ajmayan. She has also been collaborating with the Spotted Eagle Ray Research and Conservation Project at the Mote Marine Laboratory since 2016. This collaboration inspired Adi to identify knowledge gaps in research for the conservation of the globally endangered white-spotted eagle ray and to pursue her graduate studies at FAU. Her PhD thesis, Conservation Ecology of the White-Spotted Eagle Ray in Florida's Coastal Waters, addresses the lack of information about, one, their nursery grounds in North America, two, the effects of harmful algal blooms on their health, and three, the negative interactions that occur with humans and cultured shellfish in Florida. Today, Adi's presentation will focus on her research, revealing nursery areas for white-spotted eagle rays in Florida, in Florida's coastal waters, and how this is important for their conservation. So without further ado, Adi, welcome.
Thank you. Thank you for having me.
Yeah, we're so happy. We're so happy to have you for our first Save Our Seas speaker series for 2024. How are you doing tonight?
I'm doing good, thank you. I'm delighted to be the first speaker of this series, so it's exciting.
Yeah. I'm glad you're excited. Well, if you want to go ahead and share your screen with us while she's doing that, I'll remind everyone that if you have any questions, this is you can ask them. You don't need to wait to the end. Go ahead and put any questions that you might have in the chat. I'll see them and I'll ask them on your behalf for Adi. And there will be some Q&A time for at the very end of our presentation. So if you wanted to save them for then, you can, but you don't have to. You can ask them throughout, and I'll ask them for you. All right. Well, without further ado, Adi, take it away. Tell us about your research.
Thank you. Thank you so much. Well, thank you for the introduction. And hi, everyone. I'm so excited to be here today. I'm going to tell you a little bit about my Save Our Seas Foundation project that is also part of my PhD research at Florida Atlantic University. So my project for Save Our Seas is titled Assessing the Nursery Potential of Florida's Coastal Waters for the Globally Endangered White-Spotted Eagle Ray. So before I get into my actual project, I think I need to tell you a little bit about myself. So you understand my passion for sharks and rays. And so before I wanted to be a marine biologist since I was a little girl. So to pursue this career, I went to the University of Havana and got my bachelor's degree in science and biology there. This is about 2013. And then so I started working with kids at the time, and I realized that I needed to find my purpose too. So in marine biology, because I loved it all. And so I started participating in different projects involving dolphins, sea turtles, like free-living nematodes. And then the defining moment in my career was working for the Project for the Conservation of Sharks and Rays in Cuba. And at the time, I was fascinated with them, but I also was learning that they have little to no protection in Cuba. And I was also learning about the declining populations around the world. So I was really inspired by this, and I decided to pursue my graduate studies studying sharks and rays. So I started volunteer with the Spotted Eagle Ray Research and Conservation Program at Marine Lab in Sarasota with senior scientist Kim Basselshall about in 2016. And so at the time, that was my first Eagle Ray that I ever held, and that's how I also met my advisor, the Jimada Jimian from FAU Harbor Branch. And so since I was there at Marine Lab, experiencing this and collaborated with other researchers. These species went from near threatened to globally endangered. So that inspired me to pursue my PhD studying these species and finding specifically different areas related to their conservation that I could focus on to help them recover their populations. So just a quick overview about the Las Moran conservation status. They are the oldest and most ecological diverse group of fishes, and they play important ecological roles in marine ecosystems. However, even though they're so diverse, they are vulnerable to exploitation due to their slow growth, low fecundity, and the late attainment of sexual maturity, meaning they only have a few pups, and it takes them many, many years to be sexually mature. And so recently it was estimated that 1 in 4 species of sharks and rays and chimeras are also threatened by extinction. And this is due to their slow reproduction, but also they're highly susceptible to overfishing. They are taken in targeted fisheries and also as bycatch before they can even be sexually mature or even reproduce. So they're also affected by habitat loss. As an example, by the trolling of the nets over coral reefs and like the cutting down mangroves of nursery habitats that provide refuge for the little sharks and rays as they're growing. And also they're affected by pollution. And lastly, as we all know, climate change is expected to be affecting some of this sharks and rays because it's going to change the distribution of their prey over time and also their distribution as the temperature of the ocean increases. So white-spotted eagle rays sadly are not exception to this problem. So they can be found from North Carolina and the United States all the way down to Brazil. And they're also encountered in the Gulf of Mexico, the Caribbean islands, and the Bahamas, and also along the coast of Africa. So these species primarily inhabit shallow nearshore coastal habitats like estuaries and lagoons and bays and also coral reefs, and they can swim down to 70 meters deep, which is really impressive to me. And so there are targeted fisheries for these species in places like Cuba, where I'm from, Puerto Rico, Mexico, and Venezuela. And they're also sometimes they get entangled in gillnet fisheries like throughout the range too as a bycatch. So this species is sensitive to the threats because, again, they have slow reproduction. They takes them several years to be mature, and also
Mm.
um, they only have a few pups at a time. So that's why their populations are globally declining, and they have suffered a reduction between 50 to 79%, and they're now classified as globally endangered. So fortunately, this species is protected in Florida, and that's why I was inspired to uh, try to come up with a project for the conservation in Florida, because we can take measures here that can help aid with the population recovery.
Adi, can I ask you a question?
Yes.
So that distribution map, so they're all over the place, but do those populations mix, or are they local? Do they stay in one spot, or do they swim across the ocean to Africa?
They I wouldn't say all the way down to Africa, but I would say within here, within the United States, population do like there's research happening to study like the genetics of the white spotted eagle race, but
Hmm.
they do quite long migrations. As an example, from the Florida Keys all the way down to Sarasota and even more. There are cases of eagle race going from Florida to Mexico, so they do
Okay.
sometimes can cross like large distances. So
Okay.
I'm not really sure about South Africa, but they do move quite a lot.
That's really interesting.
So
So
that
yeah, so like commonly I've encountered spotted eagle rays in the shallows and on coral reefs and things. And so do they? But you said they can go to deeper water too. So they could swim from Florida, like you said, to Mexico or to Cuba or wherever, right? So
yes.
that's really interesting.
Yes, there are studies looking at this right now because we are not really sure about this long migration. So we we're starting to look more into their genetics and um, how these populations connect
Mm.
between each other. That's a really good question. So just like a general overview of this species, it's just some interesting facts. It's called the white spotted eagle race, Atobatus narinari, and that word comes from the Greek aidos, meaning eagle, and batis meaning ray, which I think is really cute. So they're known for their diamond shape and spotted pattern across the dorsal side. You probably see them uh, that jumping out of the water, or as you're like snorkeling or just paddleboarding, like or just going to the beach. Sometimes you might get lucky. So this unique pattern it's unique for each individual, meaning that it's just like our fingerprints. So no two rays are the same. So they have a combination of spots, dots, donuts, and peanuts. So the donuts are the spots with the whole center, and then the dots are fully white, and then you get peanuts, which are two dots that are connected. So this combination is like really unique for each ray. So there are studies using photo ID to look at the spots of each eagle ray to see which one it is. But
That's
it's so
really that's really cute. Can everyone let everyone look at your screen and look at that spotted eagle ray. Find the donuts, find the peanuts. Is that what you said? I see two. It's
yeah,
like two
so
spots fused together
mhm.
and
So
spots.
peanuts, donuts, and spots. And then that's really cute. And that's my favorite fact about them. But anyway, they can live up to like 14, 15 years. So we used to think they live up to 20 years, but recent studies have shown that it tends to be 14 to 15 years, and then that can probably change in captivity, but at least in the wild, that's the estimated lifespan. So for maximum size for Florida rays, the males can be up to 187 centimeters discus, so from tip of the wings. And also the females are larger, like they're 225. So there's sexual dimorphism between the sexes. So females tend to be larger because they have to carry the offspring, right? And so the size of birds that you can see here in the bottom picture, little eagle rays tend to be about 40 centimeters, and they can be sometimes even smaller, about 35, but generally they're about 40 centimeters this width, and they can have 1 to 5 pups at a time.
or have they had any studies to help identify individuals by their patterns, like you can a whale's tail or something like that, or a shark's fin?
Yes, Marine Lab is doing that kind of study. The senior scientist Kim Basishol, she's been studying that for a few years now. So they have photo IDs for all of the eagle rays that come into Sarasota Bay, and that helps them like identify which ray it is. And if it comes back, like if it tends to come back to the area, like they can compare the pictures and see, yes, it's the same ray that was here like 3 years ago.
Right. And so for everyone, I'm just gonna do a quick thing on that. I wish we were more metrically inclined, but here in the United States, we're not. So 225 centimeters, that's about 7.5 feet.
Yes.
Huge.
Yeah.
That's massive across.
It's
That's a
huge.
very large animal.
You need 45 people to even like hold the net with the rays. They can, yeah. So recently we tagged the 213 centimeter disc width ray in Sarasota, and her weight was about 300 pounds. So they can go from teeny tiny rays that are about 30, 40 something centimeter disc width to 2 meters. And
Wow.
so they can be quite large, and it's impressive. So the white spotted eagle ray is an important part of the food web because it's a prey for sharks, and it's also a predator of like marine mollusks and crustaceans. So they have a plate-like dentition, as you can see here in the bottom picture. So basically their teeth are fused dental plates, and this allows them to be durophages or shell-crushing predators. Their jaws work as a nutcracker. So they eat clams, and they have to be able to crush the hard shell, and they spit out the shelly parts. So like the parts of the shells, and they just eat the meaty parts of the prey. And then so they use at the mouth, like the teeth at the bottom, it's elongated, so it sort of works as a shovel, and it allows them to dig up some of their prey items from the sediment. They also use the snout to dig in, and they create these feeding pits when they're eating. So they use the snout to dig in, and also they can use the end of the bottom dental plate to dig up their prey. And also, like, they're considered ecosystem engineers because as they're digging out for their prey, they are known to be bioturbators. So basically, they're moving around all this sediment, allowing the nutrients to go back into the water column, and also that can fuel primary production. And also, like, as they're doing this, you can see different like fishes, like just trying to forage like around this because they're moving all of these particles in the ground, and then fish can like just eat around them, which is really cool. And then, so white-spotted eagle rays can be found in estuaries and lagoons throughout Florida, and the Indian River Lagoon here on the right. This is on Florida's east coast. And then in Sarasota Bay on Florida's west coast, eagle rays are also found throughout the area. So this is what got me like really interested about this study, because as you can see here, this is some of the cache data that Marine Lab and my lab and also FWC had for white spotted eagle rays. And as you can see here, rays from all sizes from 40 to over 120 centimeters disc width are found in the Indian River Lagoon and also in Sarasota Bay. And I just want to bring your attention to the red markings. So the red squares and the red triangles, those are little rays of for below 80 centimeter disc width. So I saw this and I'm like, these areas could be nursery areas, but I need to look further into this. And so that basically what inspired my project to look at these areas, both the Indian River Lagoon and Sarasota Bay, as potential nursery habitats for these species. So the motivation is basically, I was excited to do this project because it's a globally endangered species. They have declining populations throughout the world, but the populations seem to be stable in Florida. That's why I was like, I need to study them here. This is a great opportunity. They're also protected here. But however, we don't know, we don't have information about the nursery areas. They have not been described in the United States. And we also don't know what do eagle rays need, like in terms of like what's the preferred range of temperature, salinity, DO. We do know this about adults, but for the little ones, they tend to have a range of temperature, salinity, and other like things like do they like to be near mangroves? Do they like sandy bottoms? Do they like rocky bottoms? So this kind of information is unknown. And also the timing of places where eagle rays give birth is also a big question mark. So we don't know. We have sort of an idea when that happens, but we don't really know. So these are the main questions that that I had when I started this project, and I'm hoping to answer. So the goal of this project is to identify nursery hotspots in Florida coastal waters to reduce threats to young rays and help recover the populations. So why nursery areas? Well, nursery areas are places where they're usually coastal areas where the rays are born and when they stay until they reach maturity. And these are places where they find abundant food and protection from predators. So this is really crucial for sharks and rays, given like all the threats they have. They really rely on these areas to sort of increase the survival of their young, so to speak. Since it takes them so many years to be able to have offspring, once they have it, they want to make sure that they have a higher chance of survival. Like other species do that too. So since sergeant rays are also like highly migratory, it's really important for conservation to, instead of like trying to protect huge areas, it's base our focus on smaller areas are important for them that are considered essential fish habitats, like nurseries, feeding grounds, also like breeding grounds. So, and this is really important as an example because if you have a highly migratory species, let's say like eagle rays that go from all the way from the Florida Keys to Sarasota and beyond, you cannot protect the entire Florida coast. So you have to really find these places that are really important for the species to try to create protected area boundaries around them.
Do they prefer specific habitats for their nursery grounds? They can like the image here is like in mangroves.
Yes. So this is an Indian River Lagoon, but there's a combination because coastal development is huge in Florida. So this is, you see mangroves here, but this is not just mangroves. They're like there's a marina and bridges and docks like all around. But the preference is coastal habitats to answer your question. So
Okay.
the definition of a nursery area is like they usually like sharks and rays, or they go to coastal areas. So they go from like the open ocean, they travel to like this like uh coastal areas, could be lagoons, could be
Hmm.
bays. They are tending like most of the time are shallow. They can have mangroves. Some of the species like the lemon sharks like to be in shallow areas, like within the roots of the mangroves.
Right.
So they really want to find like these habitats that are shallow, where like big sharks cannot enter because they're too shallow for them, and also they can find refuge along the mangrove roots.
Right.
There's also a lot of food for them because
Lots
of
of shellfish they eat,
a
right?
lots of
Yeah.
shellfish. So these are very productive habitats. So that's why they typically go to shallow, like rich habitats that have a lot of food and sort of somewhat protection, like from
Right.
big
Seagrass
predators.
beds and
Correct.
mud flats and things like, yeah, that's cool.
Yeah, so like I'm still trying to figure out for with the data that I have for the little eagle rays, what it means, because it really varies. Like could be sandy bottom, could be seagrass, but that varies by location. In Sarasota, we have
Hmm.
seagrass and we have sand, whereas in the IRL, it's more like a lagoon and it's more of a muddy bottom. So like it really varies by location, but generally are shallow places, protected, it have connection to the open ocean, but they have access to the open ocean, but they can find refuge in these like coastal areas, so to speak. So how I'm planning to do this is first I've been using a catch analysis similar to the one that you saw in the maps that uses data from the Fishery Independent Monitoring Survey that has been going since 1996 to 2019. So I have quite a few data there, and then most Marine Lab data set, and also our own lab, the Fisheries Ecology and Conservation Lab. And so one of the questions that I'm planning to answer with this long-term data sets is when and where are rays born? And this is also going to provide information about the preferred range of temperature, salinity, and just like overall like type of bottom and things like that. And this data has also guide us in terms of like where to look for these rays, because initially when we started, we had sort of an idea and places where we could find them, but this has narrowed like all of the areas that we could look for. So it has been really helpful thus far.
That's a large data set too, from
Yes,
96 to 2016. Yeah.
it's very large. So generally to catch eagle rays, we use nets. So they're not like sharks that you use like hook and line and use a bait. So we use nets for them. So in the case of the Inner River Lagoon, the water is a little bit more turbid, as you can see here in the video. So you cannot, we set the nets, but we cannot visually find the ray. So it's like, and yeah, so we do sets blindly, so to speak, right? So it's basically use gill nets that are about 100 meters, and then leave the nets and then wait for the ray to go to the net. Whereas in Sarasota Bay, the water is like it's more clear and you can see eagle rays from the boats. So the methods for finding this ray, for finding rays are different in Sarasota Bay versus the IRL. Here we cannot see them, whereas in Sarasota Bay the water is clear and you can see the little ray. And then we use a seine net and we do a compass around the ray, and then we put them on the boat. We have like tanks where we put water in, and then we keep the ray there. We take a series of measurements. We take fin clip for genetics, and then we take a weight. And so these rays receive 2 different aptitude tags, which are used for aptitude tracking, which is the other part of my project. And so these tags are, as you can see here, are very small, and one of them is an aptitude transmitter that pings every second, and it lasts for about 13 days. So this is the tag that I use for aptitude tracking,
Are
meaning
these
that,
are
yes,
these radio tags or acoustic tags?
acoustic tags.
Okay, so they ping a sound that you listen for.
Correct. So they
Right.
have frequencies. It's like each tag has a unique ID. So the aptitude transmitters, the way I think about it is like they're transmitters because they transmit a signal, right?
Mm-hmm.
So the aptitude transmitter pings every second, and it lasts about 13 days, meaning that it's pinging for 13 days. So if I'm tracking for 24 hours, I can go back out, and in 13 days, I can try to find the rate over and over again. Whereas the coded transmitter, that's the one, it has a frequency of 69 kilohertz that is listened by receivers in the areas that we're studying. So, and that allows me to do long-term tracking. So one tag is for short-term tracking, and one tag is for long-term tracking up to 2 years. So this is just a quick schematic just to show you how aptitude tracking works. So it's basically following a ray from a boat using a hydrophone. So hydrophone is similar, it listens for the tags. I would call it like similar to a microphone, but not really. But the purpose of this equipment is to listen for the aptitude tag that is pinging every second. We also have a deck box and a VR100 on the boat that has a little screen and a speaker. So basically you hear the sound through that speaker. And then the way it works, it's like you have your unique ID on the screen, and you have the ping, the decibels at which the tag is pinging, and then the louder the sound, the closer you are to the animal, and the quieter it is, the farther away you are. So that's the way altitude tracking works. And I also I'm holding a compass, so the pole that I'm using has the hydrophone on the water.
Mm.
I use the compass to look at the bearing and to know where the ray is headed in relationship to the boat. So that's the definition of active tracking, which is actively following an animal from a boat. Whereas long-term tracking, you deploy the tag and then you just wait to get your data back. So those are the differences. And then so some of the questions that I'm hoping to answer with
Mm.
active tracking is what type of habitats are used as nursery hotspots, and does the habitat use and rate of movement vary throughout the day? So with this, while active tracking, I'm recording like this information about the position, latitude, longitude, and also the bearing of the ray every 5 minutes. But this equipment is recording that every second, which is really impressive. So it gives you a lot of data points, which I then use to reconstruct the track of the ray and then calculate the speed, the
Mhm.
heading, the distance, the turn angle, the net square displacement. And so this allows me to use this data to calculate the hotspots of the race. So meaning of all the area they use, which are the main areas where they spend the majority of their time, right? So we also record data about the temperature, the salinity, like throughout the day. So we do this every 15 minutes. We're collecting this data for all of the environmental variables that are
Wow.
important for
How,
this kind of study.
how fast are they? Can is it easy to keep up with them, or do they leave you in the dust?
Yeah, so it's fairly easy to keep up with little eagle rays, not so much with big rays.
Hmm.
The only times that you might lose them will be if they get spooked by something and they change direction
Ah.
completely, or if there's a lot of boat traffic. So boat traffic is really your enemy when you after tracking, because you're following noise, right?
Hmm.
And like boats make noise. So like ambient noise is really bad when you're trying to listen
Yeah.
for a tag, right? So the other times that you might be able to lose where the ray is will be like if you have a lot of boats just running by you. And something interesting about them is like they tend to spend quite a lot of time in the channels, which in the middle of the channel where
Hmm.
the boats run by. So it can be quite difficult at times.
So no,
But
so no eagle ray tracking on the weekends or like Memorial Day weekend when there's 1,000 boats everywhere, right?
no, probably no. Like we
No.
track whenever we can, but not necessarily on the weekends. But we have
Right.
had days that are really busy for tracking, but we still
Hmm.
made it happen somehow. And so this is how a track looks like. So this is an eagle ray, as you see here, the purple or the lavender line like moving down. This eagle ray was tagged in the Sebastian area in the Indian River Lagoon, and it went all the way down and then Intracoastal waterways. So that line that you see there in the map, so that's the deepest area in the IRL. So the IRL is from 1 to 2 meters deep throughout. And so the deepest area is the channel, which is 4 meters. And so that's where that ray spend the majority of the time. So that track that you're looking at, it's 25 hours. So continuously day and night. And then we relocated this ray for about 6 hours, like a few days after the initial track, which was really exciting because it showed us like they have the tendency to remain in the area for a few days, right? So they have high site fidelity.
That's really
So
cool. That's really
it,
cool to see. Yeah.
yeah, so and this is like another ray this is a different individual again, but in the Sebastian area, and you can see there like there's a bridge, so that's a Wabasa bridge, and this ray was tracked for 28 hours, sorry, and the relocation all the way down here was a week later, and we tracked it for about 3 hours.
Mm.
But the interesting thing here, like these are two rays that were tagged at different times, but they use the same areas, and like they spend the majority of their time just north of the Wabasso Bridge, which is really interesting, right? Meaning that this is an important area for them.
With this granular of data, you could probably assume, well, with more, maybe you could even assume behavior, right? So
Abs-
foraging behavior versus migration behavior, or just, swimming through behavior or whatever it might be.
Absolutely. Like for with this data, this is just preliminary. So I have in the next few months, I'm gonna be doing a lot of data analysis to actually answer these questions that I'm showing you.
Mm.
So with this, yes, you're right. We can calculate the rate of movement of the ray, and so there have been previous studies with adults where they have used the rate of movement to determine like that to calculate the rate of movement like in the channels and the deeper areas versus like shallower areas. If with that you can sort of estimate if the rate was foraging as they tend to move quite faster when they're foraging. They go to a spot and they go to a spot and then go to a spot. Whereas like when they're just resting, like they tend to the rate of movement tends to be uh less, I guess. So
Hmm.
yes, you can estimate behavior with this kind of
Well, and that tells you like what areas are, like you said, those hot spots. Like what areas do we need to protect more because it's a foraging area or it's a resting area or whatever it might be.
correct. And
That's
then
cool.
when in Sarasota Bay, you can with the adults, you're able to see like where the areas sometimes where they're feeding.
Hmm.
But yes, that's so you have the same questions that I have, right? So that's why I with the help of Save Our Seas, I was able to get some depth sensor tags, which because I was really the first tracks that I showed you didn't have information about uh where would they raise in terms of like their depth, where they're shallow, where at the surface, where at the bottom, because here at the bottom you can say it's probably foraging. So I have the similar questions that you had. So and then I obtained recently, this was just last month. We tagged actually it's May now. So this was in March. We tagged two little rays as you can see here in the picture, and it has this tag, has a depth sensor in them, and it tells you like as you can see here is that the blue line is the depth for one of the rays. And as you can see here, like in the left, you see the ray depth in meters, and you can see it by the box where they were. So this is the Fort Pierce Causeway Bridge, and this teal box is a flat. So that's a 1 meter deep, whereas the black box is a channel to this marina that is about 4 meters deep. And as you can see here, sort of like the ray sometimes in the shallow area tends to be like in the bottom, right? And it has this behavior that uh like up and down, so to speak. And then in the channel they tend to go deeper, right? Up to 4 meters deep. So this is very, very preliminary. And I still have to look further into this, but it's promising.
Mhm.
So I can hopefully let you guys know what I found out in the future. So in Sarasota Bay, we have in the Indian River Lagoon, we have tracked 5 rays, and also in Sarasota Bay, we have tracked 5 eagle rays pop again day and night. And we're hoping this will provide more information about the extent of this nursery area. So thus far, we have found similarities in these locations in terms of where are the eagle ray pops. So we were surprised that they tend to be in the same areas that big eagle rays, mainly like in the channels, and which are the deepest areas, and they're also it's also where sharks tend to be too. So that was quite interesting. And also that there tends to be they tend to have an association with bridges, which is really interesting. So we just started to find this out, and like similar to other like bonifishes, sometimes they tend to be associated with man-made structures because like it can provide potentially like refuge for them. So we're still trying to figure out why, but that's where they tend to be, in the Nursery Lagoon and also in Sarasota Bay.
That's
So
interesting.
it's really interesting. So I'm just going to tell you about passive monitoring really quickly. So if I go back, so this is the way it works. We have a receiver on the water that is a underwater listening station. And then as the rays swim by, this receiver records the ID, the date, and the time. So each tag has a unique ID number. And so they all have the same frequency, which is 69 kilohertz, and the receiver is also 69 kilohertz, and that's why they communicate between each other, because they're on the same frequency. But the tag has a unique ID, so each ray has a unique number associated with them. And then with this information, when it's retrieved, the receiver is retrieved, you can say, ray number 1213 was here this day and like at this time, this day. And then with this data, you can answer questions like, do the rays have the tendency to remain in these areas or return for extended periods? Do they use the area repeatedly for years? So, and this is important, like to know for how long do they use nursery habitats, right? So this is the way our receivers inshore are deployed. So basically we use a concrete mount and then a PVC pipe to put the receiver in, and then it has to float inshore. Most of the receiver mounts don't have a float because it's deeper, and also they can sometimes if you put them in inlets or it was too deep, they can get a tangle.
Mm.
So luckily, like these receivers are part of huge networks that are like throughout Florida. These networks are the FACT network, ACT, and also ITAG. And so we can use like thousands of receivers throughout the coast of Florida and to know when the rates go like over time, right? Which is really interesting. And also if they do leave the nursery areas at some point, where do they go? Do they go north? Do they go south? So that's really interesting.
So I just want to give you a quick time check.
Oh,
We have questions rolling in. So we have about maybe 5 minutes so we can get to some of the questions from the audience. All right.
sounds good. I'm just gonna scroll through this. So this is just to show you that we had tagged 10 rays in the Nursery Lagoon and 15 in Sarasota Bay. And then an initial analysis, as you can see here, showed that the rays tend to stay. As you can see bottom, you can see dates, and those are a few months. You can see some of our rays were here for a few months, but the data was very sparse. So thanks to Save Our Seas Foundation, we were able to acquire more receivers for the area, and also thanks to the FACT network. And this will provide more information about where these rays go. And just, I think I just got this a couple months ago, a few months ago, and it's really interesting to me. One rate that was tagged here in 2022, like it's has stayed in the IRL for over a year now. And as you can see here, if you match the dots, the color dots, you can see this ray went up and down in the Indian River Lagoon for a few months now, which is really interesting, meaning that they do stay within the lagoon for a considerable amount of time. So I had to look further to see if they do go out and they come back in, when do they do that? And also in Sarasota Bay, we have acquired quite a few data from, these are 5 pups that we tagged, but right now we have 15, which is really exciting. And so here you can see all the dots are detections from these animals. And at the bottom, you can see dates, like showing also that they tend to stay in these areas for a few months. So, and if they do come back out, which are the different colors, they are not, that the areas where they were tagged are pink and orange. So the other colors meaning that they went to different areas, but they did come back to the same spots, which is really interesting and it's promising. And I think with that, I just want to thank Save Our Seas Foundation for the support of this research. Also my lab, because I couldn't do this without them, and all the collaborators and researchers that have helped me with this. I would like to know if you see white spotted eagle rays in the Inner Lagoon, Sarasota Bay. I would love to hear from that, have a picture, and just to know where the little rays are. And that's my contact information. And I have this slide. This is one of your questions. I don't know if you want to ask me that. I
Yeah,
just,
go ahead. How
I
can we get
just
involved? How can we help?
yeah, so you can get involved by the most important thing and the main takeaway from this talk is to spread awareness about eagle rays and how beautiful they are and how we need to protect them. And so that's the main takeaway. If you can spread awareness and just share it to your friends and your family, that will be greatly appreciated. Also get to know us. You can follow us on social media, our lab, the Fisheries Ecology Lab. That's the Pacheco HBOI, and also my social medias are in Nari. I post cute pictures of eagle rays. So if you're interested, you can follow me, and also that's my email. If you have any questions, you can email me. I would love to see or know where you see eagle rays. And then so also you can visit the Ocean Discovery Visitor Center, our harbor branch, and learn from our Ocean Series lecture. We have different scientists talking about all kinds of research, and also become a volunteer if you're interested in this. And with that, thank you very much. That's
Oh,
my
thank
talk.
you. No, it's beautiful. I being in South Florida for a long time, I've spent a significant amount of time on the water, kayaking or paddleboarding, and I guess lucky that I didn't realize they were endangered worldwide, but you see them quite frequently here in South Florida.
Wait,
Which is amazing, and it's always a great day. You know, it's going to be a great day when you see an eagle ray swim by. Um, so well, thank you very much for sharing your wonderful research and what's happening. Like you said, it's still ongoing, so we'll have to have you back, I guess, right? To give us an
yeah.
update.
Yeah, it's still ongoing. I'm on my third year and I'm planning to graduate next year. So I just tagged all of these rays and I'm just starting to get data back, but I wanted to just give the talk just to show like some preliminary data we have and some promising
Mhm.
results that these rays do tend to be in these areas, and it seems to be important for them. So in the next few months, I'm going to continue doing more data analysis, and I can hopefully share it with you guys next year.
Yeah, that'd be
Hopefully.
great. We'd love to see it. Um, well, great. So we do have some questions from the audience. One question we had is can you what there's a lot of places in Florida in general, excuse me, that you can find rays in captivity, like cow nose rays and these kind of things, in aquariums and whatnot, or yellow rays or things like this. Are there any places you can see spotted eagle rays in captivity that you can go and observe and admire their beauty? Do you know of?
Um, yes, in Florida, I'm not so sure of. I believe that the Frost Museum of Science in Miami might have.
Oh, and frost. Okay. Yeah.
Yes,
Are
I'm
they
not.
easy to keep in captivity? Well, easy. Not for like a person in your house, but for an aquarium. Is it a common thing to see them?
Yes. So
Hmm.
I have seen him at Georgia Aquarium. I have seen him in Tennessee Aquarium and an aquarium in Tennessee. I have seen him in several aquariums.
Hmm.
I will say yes, once you know, once you an aquarium, yes, you can once you know their nutrition, what they
Hmm.
need in terms of
Of
water
course.
temperature and things like that. Yes, they're fairly easy to
Yeah.
keep in captivity.
Well, and based on your data, we know that they like to swim a lot, right? So
Yes, correct.
they need lots of space. Cool. Let's see. So we had some interest just in you as well. Thank you very much for sharing a little bit about yourself in the very beginning. And how did you come across this? How could someone follow in your footsteps? It was more of the question. For anyone young or older that's thinking of a career in marine science, what would be a piece of advice for them to learn more and to follow you?
I would say find something that you're passionate about and just run with it. Because that's yeah, if you have passion for something, like you're gonna find like the perfect project for you, and
Mm,
I wouldn't say like don't give up. So sometimes it's really hard to find like the perfect niche like for you, like the perfect school to go to, like should I go to this program, should I go to this one, and it's just all about like being persistent, and
mm.
as I would say, if you keep going, like everything's gonna line up for you at the right time, I guess. But
Yeah.
it's just as long as you really want to do something, you put your mind on it, it's gonna happen
Yeah,
no matter what.
follow your passion, right? That's a great piece of advice. Do something you like doing, right? We
Correct.
can
So
I can tell you really enjoy what you do. So,
it's I mean, it's like with to work with like baby eagle rays. Who doesn't like baby eagle rays? So,
right?
but I do other things too, and I like all of them. It just yeah,
Yeah.
find something
That's
that you really like, and once you do that, like it doesn't really feel like a job.
yeah,
You
right.
know?
Yeah, that's that old saying, right? If you love what you do, you don't work a day in your life or something, right?
Yes.
Um, well, so I think I kind of see where your project is going, and how you might make it or bolster the data that's being collected and things like that. Um, but what would you do if you were, to take it to the next level? What's your on your wish list, right? If someone were to give you that blank check, you never know who's listening. Um, what would you do to make your project bigger and better and do more research?
To make my project bigger and better, I think this type of project like related to acoustic telemetry, it's quite expensive. Like the receivers, the tags, everything. Like it will be get to spend like the array of receivers specifically like in the inner
Mm,
Blue Lagoon, and I would also like to my dream will be to expand this kind of research to like places like Bermuda and the
mm,
Bahamas, where this species is also very common and protected. Um, so the main goal of my research is to provide information like for management and conservation of the species, right? To sort of know what the most important areas for them and try to protect them and like a little bit more, and then to see if that helps with the recovery of the populations worldwide. So
right.
like there are not a lot of regulations in other countries, like not a lot known in Colombia and in Mexico and Venezuela, so the regulations there are not as strong as ours, so that I'm hoping to do more in the places where they are protected. So hopefully, like, the populations increase a little bit more, like if we do that in this region. So my dream will be to expand this kind of research like beyond Florida if
Mm.
I can.
Yeah. Those, you mentioned those acoustic networks. Do those networks exist outside of Florida? Like you mentioned in some of the Caribbean islands and things, those acoustic long-term monitoring, those projects are there too.
Yeah, so Ocean Tracking Network manages thousands of receivers like all
Mm,
over the world. So like there are different arrays in different countries. So and if they're not, like they can like the same way we did, we created our own
yeah.
for this research. So that can be easily done.
Yeah. To
Yeah.
expand the network, get more data. More data is always good, right?
Yeah.
Well, I think that's going to run us up to our time today. Thank you, Adi, very, very much for coming and sharing your passion about white-spotted eagle rays and letting us get a little glimpse into what you do. Thank you very much.
Thank you for having me. I'm always
Yeah.
happy to talk about eagle rays.
And like everyone, like she said, if anyone has any questions, feel free to reach out. Send Nadi your pictures of spotted eagle rays if you
Yes,
see them
please.
with location data, probably be important too, where you saw it, when you saw it.
Yes, that'd be ideal. And like day and like a location. Doesn't have to be latitude, longitude, but a location would be great.
Yeah. Well, thank you again very much. And again, thanks to Save Our Seas Foundation for funding your research and making this series possible. Join us next time. Excuse me, as we have our next speaker in June, Isa Seydu, about protecting sharks in Ghana. So we're going to Africa next time. And I think it'd be a great conversation. Same time, 6 to 7 p.m. Eastern on June 5th. You can go to mods.org/STEM Learning Series to sign up and register and meet us there. All right. Well, thank you again, Adi. It's been a pleasure. Hope everyone has a great evening.
Thank
Bye.
you. Bye.